Hello everybody, thank you so much for your time. My name is Suhair Khan and today we'll be looking at a possible mechanism for cognitive resistance to neurodegeneration, and that is through modulation of interneuron activity in the hippocampus after exposure to enriched environments. I would first like to discuss what area of the brain we'll be looking at in today's research. We can focus our attention down below here to this yellow figure. This here is the hippocampus. We will be focusing on the dorsal hippocampus, which is seen here in the top left. And the dorsal hippocampus is involved in functions such as maintaining your spatial awareness, as well as being involved in memory making and long-term memory and other cognitive functions. Now, before I get into my data or even my hypothesis, I first want to talk about what inspired this research in the first place. And I will do so by reciting a quote said by Dr. Kageyama himself. What's better than a cure for Alzheimer's disease? Using preventative measures to delay or prevent the development of onset symptoms of Alzheimer's disease. Throughout your life, all the physical activities combined with cognitive activities you have engaged in help develop what is known as your cognitive reserve. Your cognitive reserve is your brain's ability to cope with brain damage. And as is illustrated here in this graph, an individual with a greater cognitive reserve has the ability to resist the decline in cognitive status once presented with a neurodegenerative disease, in this particular case, Alzheimer's disease. Your cognitive reserve can be strengthened by engaging in both physical and cognitive activities. The brain does not birth new neurons after you are born, except for a few exceptions. One area that maintains this ability is the dentate gyrus of the hippocampus, and the new cells that are born here are known as newborn dentate granule cells and are stained here in green. Generally, a large portion of these newborn cells don't survive. However, with an enriched environment, as can be seen here on the right side, a greater number of neurons are born, as can be seen here in green, and a greater number of neurons are able to survive and integrate themselves deeper into the circuitry of the hippocampus. It is now established that an enriched environment leads to an overall greater number of neurons in the hippocampus. This means that there is a new and increased demand for energy from all these new cells. If proper energy demands are not regulated, this will lead to hyperexcitation of the hippocampus. This particular work of research shows that hyperexcitation leads to the death or the loss of interneurons, which ultimately leads to dysfunction of the hippocampus. On the left is a control brain, and on the right is a hyperexcited brain. You can see that in the hyperexcited brain, there is a significant decrease in the number of interneurons, and the mechanism that they proposed was due to a deficiency in dendritic inhibition, and they measured that by counting the number of symmetrical synapses along the axo-initial segment. The hyperexcited brain, which is here on the right, had significantly less symmetrical synapses, indicating that proper inhibitory signaling is necessary for proper hippocampal function. Indeed, if inhibitory interneurons are necessary for proper hippocampal functioning, then it is imperative to determine what mechanisms control these inhibitory interneurons themselves. And that leads me to my hypothesis. If rats are exposed to an enriched environment, then there will be an increase in the number of NADPH-DIAPHORASE interneurons to compensate for the increased demand of energy. We chose to look at NADPH-DIAPHORASE positive interneurons because they are cheap to look at, and additionally, the majority of them are GABAergic, meaning that they are inhibitory. This particular cell here is known as an Ivy cell, and as you can see, it has thin varicose dendrites that extend outward into the granule cell layer, inhibiting the activity of these dentate granule cells, making them ideal targets to look at when researching cognitive reserve and other neuroprotective agents. We had three separate groups. The first group is the control group, and this group of rats received no forms of enrichment. The second group is the enriched environment group, and this group had a cage that was double the size, and they also received toys to play with, and these toys stayed in the same positions and stayed stagnant for the entirety of the research duration. The third group is the enriched changing group. The enriched changing group had a cage identical to the second group, However, the locations and positions of these objects were rearranged 3 times a week. They were swapped up for new novel objects once a week, and on top of all of this, the rats were allowed to enter a play arena 3 times a week for 4 hours, meaning that these rats were allowed to go to Disneyland for 4 hours 3 times a week in order to stimulate as much enriched environment as possible. And then what we did is we divided the hippocampus into its 7 respective layers, and we individually counted each interneuron in the respective layers. And this took a very long time. I'm very grateful to every single person who helped participate in this process. We were then able to accumulate this graph, and we saw that in 6 out of the 7 groups, there was an increase in both enrichment groups of the total number of neurons. Additionally, we then standardized this number and still saw the same results, that in both enrichment groups, there is an increase in the total number of NADPH-DIAPHORASE interneurons, even in the standard of per millimeter of dentate gyrus. Although this alteration in neuron number is potentially compelling, we recognize that our samples are relatively small, and this data should be recognized as preliminary. Regardless, these preliminary results are still very significant because they highlight a new mechanism of physiological homeostasis as well as a physical evidence of a cognitive reserve existing. And that mechanism is that due to the increased demands of energy that are derived from newborn dentate granule cells, the brain will regulate its energy expenditure through an increase in the number of interneurons or the activity of interneurons. And this is important because this has very rarely been shown in literature, as most works of literature focus on restoring post-lesion levels to the control level. We have shown an increase from the control level to a new increased level, showing that the brain has the ability to put armor on. Some of our future work includes looking at these calcium-binding interneurons, as well as looking at different regions of the brain of the hippocampus, such as the CA1 and CA3 regions. Overall, in today's presentation, I've discussed that you can increase your cognitive reserve by engaging in physical and cognitive activities, and this is necessary in order to maintain hippocampal homeostasis. We induced new energy demands by presenting rats in enriched environments. The rats adapt to this by increasing the number of NADPH-DIAPHORASE interneurons, and this is important because it shows physical evidence of a cognitive reserve and a new mechanism of physiological homeostasis that could prevent people from potentially developing neurodegenerative diseases. Thank you so much to everyone who participated in this research. Thank you, Dr. Kageyama. Here are some of my references, and I am now open to questions, and if you'd like to contact me, here's some of my information. Thank you so much.